Hello and welcome to the IB revision channel. Today we're going to be looking at 1.2, which is the ultrastructure of cells. So to begin with, our essential idea is that eukaryotes have a much more complex cell structure than prokaryotes. So an essential definition for this topic are organelles. So organelles are specialised substructures within a cell that serve a specific function. And two of the universal organelles are ribosomes and cytoskeletons, which are found in both prokaryotic cells and eukaryotic cells. An essential IB requirement is to be able to draw and label a diagram of the ultrastructure of E. coli as an example of a prokaryote. The diagram should show the cell wall, pili, flagella, the plasma membrane, cytoplasm, 70S ribosomes, and a nucleoid with naked DNA. So as well as this, we should be able to annotate the diagram with the functions of each named cell. So prokaryotes have a simple cell structure without compartments. They were the first organisms to evolve on Earth and remain to have the simplest cell structure. Along with that, they can be found almost anywhere, so in the soil, in water, on our skin, in our intestines, and even in pools of hot water in volcanic areas. So all cells have a cell membrane, but some cells also have a cell wall, perimeting the plasma membrane, and this serves as an extra protective shell around the prokaryote to protect it and prevent it from bursting. As no nucleus is present in the prokaryotic cell, its interior is completely filled with cytoplasm. Along with that, the only other organelle that prokaryotes have are 70S ribosomes, and these fill up the inner cavity of the cell. However, some areas on micrographs may seem lighter, and this region contains the DNA of the cell, which is usually in the form of one circular DNA molecule. This DNA is not associated with proteins, which explains the lighter appearance compared with other parts of the cytoplasm that contain enzymes and ribosomes, and this lighter area is called the nucleoid. So prokaryotic cells do divide in their own special way, but instead of dividing through mitosis as eukaryotic cells do, they divide by binary fission, and it is used for asexual reproduction. So the process of binary fission is that the circular DNA is copied in response to a replication signal, the two DNA loops attach to the membrane, and the membrane elongates and pinches off, known as cytokinesis, which forms two new cells. So here's a visual diagram of the process of binary fission, and I've just summarised it into three main points, so copy, attach, and elongate. So eukaryotic cells have a much more complicated internal structure than prokaryotic cells, and an IB requirement is to be able to describe the eukaryotic cell and label it. So whereas the cytoplasm of a prokaryotic cell isn't one undivided space, eukaryotic cells are compartmentalized, and this means that they are divided up by partitions into compartments, and these partitions are single or double membranes. The most important of these organelles is the nucleus, as it contains the cell's chromosomes. Each organelle in the eukaryotic cell is specialised to perform a specific role. So here are the functions of each of the organelles. You can just pause it here and take a look at the functions and try and remember them. The diagram should show the plasma membrane, the cytoplasm, 80S ribosomes, the nucleus, mitochondria, rough endoplasmic reticulum, Golgi apparatus, vesicles, and lysosomes. So here is a plant cell, and it's different to an animal cell because it contains chloroplasts and a cell wall. Here are the functions of the organelles in a plant cell. So we need to be able to interpret electron micrographs of eukaryotic cells to identify features which are listed above. So there are a few differences between electron microscopes and light microscopes, and this includes that the electron microscopes have a much higher resolution than light microscopes and a much higher range of magnification. So to remember these, they're just two key points, so high resolution and high magnification. The two main types of cells are prokaryotic cells and eukaryotic cells, and they have some vital differences which we need to be aware of, and these can be remembered by DNA, organelles, reproduction, and the average size, and it can be remembered by the acronym of DORA. So prokaryotic cells have naked DNA, whereas eukaryotic cells have circular. Prokaryotic cells have 70S ribosomes, eukaryotic have 80. The reproduction is binary fission compared to mitosis and meiosis, and the average size ranges from 1 to 5 micrometers to bigger than 10 micrometers in eukaryotic cells. That is everything that we need to know for 1.2 biology, the ultrastructure of cells. Please make sure to like the video, give us a comment, subscribe, and turn the notifications on if you want to see more videos. Thanks a lot. Bye.